Okay, we're going to continue the rest of Chapter 3. The beginning in the previous presentation we just looked at cells in general, and now we're going to look at some more detail in a eukaryotic cell. And we're going to start out with looking at the different types of organelles. Okay? To remind you, the definition that we gave a moment ago is that an organelle, by the definition that we're going with, is that these are going to have a membrane around them. They're going to have a little phospholipid bilayer around these structures that are within a cell. Okay, so they're separate structures, they're membrane bound, compartmentalized within the cell. Okay. One important organelle we've already identified is the nucleus. We'll study its structure in better detail. We're going to talk about both the smooth and the rough endoplasmic reticulum. We'll also talk about the Golgi Complex, or you can call it the Golgi apparatus, or even the Golgi body, Lysosomes, Peroxisomes, Mitochondria, and then we'll look at some specialized features in plant cells, including a central vacuole and chloroplasts. Starting out with the nucleus, we have an idea about the nucleus. We know it's there to compartmentalize DNA. It regulates other cell functions. It's besides just containing the DNA, the nucleus is much more important to that. But we'll look at it just from containing the DNA and what happens inside of the nucleus. But basically, the nucleus is sometimes referred to as like the brain or the control center for the cell, and that's what it is. It regulates that cell's activities. The structure of the nucleus is kind of complex. And it makes sense because the nucleus is kind of really important. And so it does have a membrane around it. However, it actually has two membranes around that. Looking at my note that I have here for you though, some cells, most cells, eukaryotic cells, are mononucleated. That means they have one nucleus. However, some tissues have developed so that they are multinucleated, which doesn't seem logical. However, when some organisms' tissues grow, some cells actually fuse, like 2 cells or 3 cells or however many cells might actually fuse together, and then therefore the nuclei of each of those cells are all within what would be a whole single cell. So some cells can actually have multiple nuclei. In your body you have some cells like this. For example, in your skeletal muscle are multinucleated, as well as so are your cardiac, the cells in your heart, they're multinucleated. Also, some cells are anucleated, which doesn't make sense because I just got done telling you that all eukaryotic cells have a nucleus. And then this next statement is saying anucleated. Some cells lack a nucleus. This is also a developmental feature. A classic example in your body, that's your eukaryotic organism, and you do have some anucleated cells. You've heard of your red blood cells? When your red blood cells grow and develop, they actually eject their nucleus, so they can be small enough to circulate through your blood, which, if you think about it logically, without the nucleus, the cell can't divide, which makes sense. Why you hear all the publication why it's important to donate blood, because our blood cannot reproduce itself. The mature red blood cells cannot. If we look at the overall structure of the nucleus, it does have a nuclear envelope. I want you to make a note about the nuclear envelope, as I alluded to a minute ago. The nuclear envelope is double membraned. That means it has 2 phospholipid bilayers around it. If you look fairly closely, over here in the far right hand side of the image, if you look really closely, you can see in the purple is the nucleus. One, and then right next to it, you can see really closely a second membrane around it. So not only does the nucleus have a membrane around it, it actually has 2 membranes around it. So the nuclear envelope is double membrane. Additionally, the nuclear envelope has these things called pores, and as the name pore implies, it's an opening, and therefore that tells us that products can enter and exit the nuclear envelope. At the end of the semester, I think in the third unit, we'll talk about what does and what does not enter and exit the nuclear pore. But they're there so that the nucleus is not completely shut off from its cytoplasm, from the cell's cytoplasm. In addition to being double membrane bound and having pores, if you look on the dark nucleus in this image, you can see the little golden dots. Those are ribosomes. So the nuclear envelope also has ribosomes attached to it. Okay? So in summary, the envelope structure is complex. It's double membrane, it has pores, and it also has ribosomes attached to it. Okay. So that's the nuclear envelope. Another important feature of a nucleus is the innermost compartment of the nucleus, and it's called the nucleoli. If you see it with the suffix I, that means there's more than one. If you see the suffix us, nucleolus, that means there's just one. And if you look at the image to the right hand side here, I can see a single nucleolus. It's easy, if you're looking at something under a microscope, electron microscope, it's easy to maybe confuse the nucleolus from the entire nucleus. And one way to differentiate it is that the nucleolus does not have a membrane around it, so that tells you it's inside of that double membrane nuclear envelope. What the nucleolus does is help make ribosomes, right? Everything has to have somewhere that it gets created from, and the ribosomes get their manufacturing, part of their manufacturing here through the molecules in the nucleolus. And the third feature of the nucleus is the chromatin, and the chromatin in this image are the kind of stringy looking purple fibers. What chromatin is the DNA, but it's not just the DNA. It's DNA with these proteins that kind of organize it or package it inside of the nucleus. Okay, so the chromatin is basically the cell's DNA, and you can see how it's dispersed throughout the cell. Your textbook has some nice images of the nucleus. Again, the nuclear envelope, we can appreciate its double membranous by the yellow. You can see the two lines. You can also see the pores and how they kind of form channels or pores, so allow for entry and exit of materials. What is not shown in this image is the presence of ribosomes. Okay, in this. If you look at the right hand side, there's a nice transmission electron microscope image, or TEM image, that has been colorized. The yellow boundary on the far right hand side is part of the chromatin, and then you can see the little spaces in between. That will be the nuclear pores, and then right in the middle you can see that purple stained area, the nucleolus. Okay, so a nice little image of a nucleus. Okay, another important organelle to eukaryotic cells is this thing called the smooth endoplasmic reticulum. It gets its name because, in contrast to the rough endoplasmic reticulum, it under a microscope looks smooth. And in this image, on the lower right hand side, you can see the smooth endoplasmic reticulum in the back end. Okay, so I'm going to try to highlight it in this orange color. Okay, so this whole tubular looking organelle, you can see how these little tubes are all interconnected. You can see that they are definitely membrane bound, and if you can kind of imply that it forms kind of a hollow something in the inside of it. Okay, it's not just hollow, there are enzymes that go to work in there. And collectively, what the smooth endoplasmic reticulum does is help manufacture lipids. Remember, we studied those in Chapter 2. They have to come from somewhere. We talked about cholesterol can be synthesized in the body. Cholesterol, your hormones, estrogens, etc. Get their beginnings here in the smooth ER, as well as your triglycerides, phospholipids, etc. They get their orientation here. Some cells are specialized for their smooth ER to actually store ions. For example, in your skeletal muscle in your body, or your heart, or your smooth muscle in your stomach, your digestive system, those cells, smooth endoplasmic reticuli, actually store calcium. ion in there, and then regulate calcium flowing in the cytosol and back into the smoothie yard. So smoothie yard is a dynamic little organelle. As implied here in this image, it's generally associated with around the nucleus. Okay, so you can see the smoothie yard, and it's adjacent to the nuclear envelope. If we look at its structure, as we implied, it is tubular in nature, and it's got phospholipid bilayer around it, but it does not have any ribosomes attached to it. Okay, that's the smoothie yard. If we contrast that to the rough endoplasmic reticulum, the rough endoplasmic reticulum is also usually actually connected to the nuclear envelopes. It's usually an extension. You can kind of see that here where the purple nuclear envelope is branching off into these flattened sacs of the rough ER. So I'll highlight the outside of the rough endoplasmic reticulum. Okay. You should also notice how in this image little golden ribosomes are attached on here for the rough endoplasmic reticulum. You should also be able to see the inside. Okay, so the same thing inside of the rough ER are enzymes that go to work. And what the enzymes do for the cell is help make proteins. Okay, I want you to take a minute here to really differentiate what kind of proteins are made here. The type of proteins that are made at the rough ER are those proteins that are going to be secreted out of the cell. Like, I'm going to go back with insulin. Insulin's a protein hormone, so it's got to be made inside of a cell and then released out into the blood. As well as we're going to talk about membrane junctions. Right? Those proteins have to get made somewhere and then released outside of the cell. In those, when we talked about the membrane earlier, we talked about some proteins can form channels or aid in cell-to-cell recognition for glycoproteins, etc. Those types of proteins get their origin from the rough endoplasmic reticulum. Okay. What I want you to really understand is that the rough ER does not make all of the proteins for the cells. It makes only those proteins that will be secreted out into the environment, okay, like hormone or something like that, or actually incorporated in a membrane, okay, like in the cell's plasma membrane. Okay, those are the jobs of the rough endoplasmic reticulum. If its basic structure, it's flattened membrane, and it is covered with ribosomes. I want to draw your attention on this image for the rough endoplasmic reticulum. If you look on the very lower right hand side of the rough ER, you can begin to see where the little rough ER is beginning to bulge off and kind of pinch off. And what happens is when proteins are synthesized, they've got to get out of the rough ER. And the way that they get out of the rough ER is the rough ER actually pinches off a part of its membrane, and so that the inside of the rough ER, the content of the newly synthesized protein, is now separate in its own what we'll call a vesicle. And it's going to be transported. Okay, where it goes is to the next organelle called the Golgi Complex, or the Golgi body, or the Golgi apparatus. So before we look at it, I want to draw your attention to the image on the lower right hand side. If you see the little pink sphere, that's representing the membrane that was just pinched off from the rough ER. Inside of it would be some newly made or newly synthesized proteins. The yellow and the green are just representing some proteins that were just made. And so you'll notice that those proteins are no longer trapped inside the rough ER. They're now trapped in this vesicle. And you'll notice that if we time phase forward, that vesicle then moves over towards the Golgi apparatus. And so what the Golgi does is takes those proteins that were just manufactured in the rough ER and is going to modify them. Okay, types of modifications that happen would be, for example, glycoproteins. The protein is made in the rough ER, but the sugar, the carbohydrate group, will be added on here in the Golgi complex. So that's what the Golgi does. You'll notice that its structure, I'll come back to that statement in just a minute. You'll notice that its structure is also, it's a membrane-bound structure. It's really flattened looking, and it's proximal to the rough ER because it's working very closely with the rough ER. Going back to the note, the little gray box that I overlooked just a minute ago. It does manufacture other membrane-bound organelles, including the lysosomes and the peroxisomes that we'll address in just a minute. The proteins will enter the Golgi complex, they'll be dumped into the Golgi complex, and then processed through. And if you look on the opposing face of the Golgi complex, you'll notice that the proteins are now, can you see how they're no longer mixed up? Proteins of the same type get grouped together and then packaged into a new little vesicle. Okay? So it's just kind of a processing plant, if you will, for our proteins. Okay? They come in from the rough ER in a vesicle, get processed in the Golgi, sorted off, and then repackaged in a new vesicle. That vesicle may be incorporated into the cell membrane, or secrete those contents out into the out in the extracellular fluid outside of the cell. Or they may temporarily hang around in the cell to work as these other organelles called lysosomes and peroxisomes. The lysosomes, if you look at this image, we can see the phospholipid bilayer, and the image is overlaying a transmission electron microscope image. And so even without knowing much, we know that the lysosome was modified in the rough ER, so that tells us, modified in the Golgi, so it must have been manufactured in the rough ER, so it must have proteins inside of it. Okay, and it does. It has proteins inside of it, and what it does is help break down foreign substances, like maybe if a cell interacts with a bacteria, the bacteria could be broken down through the action of the lysosome. Or maybe a mitochondria. It's getting too old. It can get broken down by a lysosome. Okay, the way it does this is because it has inside of it these enzymes called hydrolases. Okay, so I want to take us some time and walk through this word, hydrolases. Anytime you see this suffix right here, ASE, that tells you that this thing is an enzyme. Okay, an enzyme. We haven't studied enzymes yet, but basically they're proteins, which makes sense, it's falling together, and they help speed up chemical reactions. Okay, so ASE suffix tells me this is an enzyme. And this class of enzymes are called hydrolases. Okay, so looks like there's going to be a few interruptions between the kids this morning, but I'll try to pick up where I left off. A lysosome, I think I was talking about enzymes. The suffix ASE indicates that this is an enzyme, and then we understand fundamentally that enzymes are proteins. This particular class of enzymes that you'll find inside of a lysosome are called hydrolases, and they hydrolyze or form hydrolytic reactions. And if you recall from chapter 2, those types of reactions break down chemical bonds. And so these hydrolases break down chemical bonds to make substances non-toxic to the particular cell. Okay? Once they break something down, like a bacteria or an old organelle or whatever the case may be, it would then actually purge those destroyed contents back out into the extracellular fluid. So it's out of the cell and not toxic or damaging. Those are called lysosomes. There are some very similarly built structures inside of a cell, and they're called peroxisomes. Peroxisomes are also just membrane-bound vesicles. They have inside of them enzymes as well, which implies that they're proteins, which implies that they were processed in the Golgi, which implies that they would have been manufactured at the rough ER. Okay? And what the peroxisomes do is they also have this kind of detoxification type process. Generally, peroxisomes take on like alcohols in the environment or other lipids and breaks them down into non-harmful substances. Okay? Fats. Okay. If we look at the basic structure, it too is just a vesicle inside of the cytoplasm, inside of the cell environment. And if you look, it also has a class of enzymes called oxidases, and then a second class of enzymes. What the enzymes, oxidases, do is when they break down fat products or alcohol-containing products it produces something called hydrogen peroxide, or is illustrated here, H2O2. You've probably heard of hydrogen peroxide. You've probably seen it. You maybe used it before. And you notice that hydrogen peroxide, when approached to something that's like really reactive and bubbly, inside of a cell, that's not good. So we have a class of enzymes called oxidases that actually produce hydrogen peroxide, which seems counterproductive. Okay, well, what happens is hydrogen peroxide is a byproduct when we catabolize different waste products. Okay, since hydrogen peroxide is toxic, potentially toxic, what happens is there's not just one, but two classes of enzymes inside of the cell. The other class is called catalases. We can still see that these are enzymes, and what they do is they take that newly produced hydrogen peroxide and produce it into something not so toxic, including oxygen and water. Okay, so oxidases will break down the initial toxic product, producing hydrogen peroxide, but as soon as it's produced, there's a whole other set of protein enzymes in there called catalases that break the hydrogen peroxide down into oxygen and water, making it, right, obviously not toxic. Okay, those are peroxisomes. Another important organelle that we'll study include mitochondria. I'll study or spend a little more time on the structure of the mitochondria because we'll definitely study their function in, I think this coming up in the second unit. And what they do is produce for a cell abundant amounts of ATP. Tons of this molecule called ATP. And for right now, you can just say that's a molecule for energy transfer. Okay, so it helps synthesize a usable form of ATP for a cell. It uses or carries out two important processes, including the Krebs, or I think your book may use the citric acid cycle, and another process called the electron transport chain. So coming up this semester, we'll study these reactions in great detail, the Krebs cycle and the electron transport chain. The way it does this is due to the way it's built, because if you remember, structure dictates function. And of course, we've got it in our list of organelles, and so it's got to have a membrane around it, but this one also has two membranes like we saw with the nucleus. The mitochondria are also double membrane structures. If you look at the image on the right of a mitochondria, the outer membrane is kind of continuous and smooth looking, the brown looking feature. Okay, so I'll try to highlight here. Okay, that would be the outer membrane. And then there's also an inner membrane. Okay, the inner membrane I'll try to draw here in green. Okay, notice how the inner membrane actually folds. You see how it bends and folds. Okay. Anytime something folds in an environment, right, in a biological system, it's there to increase surface area. And so basically we can get a lot of chemical reactions in a small little compartment, right, going on. Additional areas that I want you to identify, so maybe you want to on your own draw the mitochondria, but I want you to identify the area that's colored here blue as the matrix, the mitochondrial matrix. That's where the Krebs cycle and the electron transport chain are going to occur. Okay, so we're identifying the outer membrane, the inner membrane, and the matrix. Okay. You'll notice that there's one more area that's not labeled in this image, but we can't not talk about it, and it's the space in between the two membranes. I'll try to color this purple. Not working out too good. And the space in between, okay, the outer and the inner membrane. It looks void here, okay, but it's certainly not a devoid area. It's fluid filled as well, and I want you to label that as the intermembrane space. The space between the outer and the inner membrane is called the intermembrane space, and you would spell that I-N-T-E-R, intermembrane space. Okay. All right. We identified the outer membrane as smooth, the inner membrane does fold to increase surface area. And something really interesting about the mitochondria is the mitochondria actually has its own DNA and its own RNA. So many of its proteins that it has expressed inside of it, carrying out the Krebs cycle and the electron transport chain, it actually makes itself.
Shelter?
Okay. So mitochondria, in summary, right, it has two membranes. The outer membrane is smooth, the inner membrane folds. The innermost fluid-filled area is called the matrix, where the Krebs cycle and the electron transport chain happen. And there's a space in between the outer and inner membranes called the intermembrane space. And as well, the mitochondria is unique in that it has its own DNA separate than the nuclear DNA. Okay. If we look at some other specializations, some cells have vacuoles. Plants have what's called a central vacuole, which you can see in this image over here in the big area right here. So we've got it under the category of organelle, so it tells us it has a membrane around it. And it's this great big giant fluid-filled vat. In the plant, what it's used to do is store nutrients, okay, for the plant. It may have some catabolic ability to break down the substances, and it certainly makes sure the pH is in balance within the cell. So vacuoles are more characteristic with plants. Some animal tissues have vacuoles, but they're not big and massive like this. They're tiny, like the lysosomes and the peroxisomes. Okay, but so from the standpoint of your book is looking at the large central vacuole. Okay, so I identified it's larger in plants and not very obvious in animals, or they're much smaller and more numerous in animals if they're present. And again, nutrient storage, waste, breakdown, pH balance, there's a whole bunch of type of chemical reactions that can occur inside of a central vacuole. Another structure characteristic of plant tissue are these things called chloroplasts. And what chloroplasts do or allow for a cell to carry out a process called photosynthesis, which you've probably heard the word before, which allows for cells to capture light energy and produce food. This is what allows for some organisms to be phototrophic or autotrophs, right? Make their own food. Chloroplasts are most synonymous with plant tissue. However, you'll find them in some fungi, in some protists. Some protists have chloroplasts. Some eukaryotic organisms have chloroplasts as well. But from our exposure, we could just kind of be safe and just say plants have chloroplasts. We understand that plants can capture sunlight. Chloroplasts do have the ability to store some starch, so we can not only just carry out photosynthesis, but also store some nutrients. Like the mitochondria, I want to spend some time on chloroplast structure because we really need to understand the structure of it to understand them better its function. And again, I think that's in Unit 2 where we're going. We'll look at chloroplasts, maybe Unit 3. But the chloroplast is also double membrane. So if you look in the image on the far right-hand side, you can see one highlighted in purple and then two. You'll notice that the two membranes are smooth in appearance, and there's a space between as well. But technically, there really are three membranes inside of a chloroplast. Okay, if you look right in here in these dark green areas, okay, where my arrow is, those little stack looking things are called thylakoids, and they are also membrane bound. So technically there's 1, 2, 3 areas of membranes inside of a chloroplast. Okay. The thylakoids that are stacked in there contain the molecule called chlorophyll, which allows for the photosynthetic reactions to take place. Okay. Like our chloroplast, like our mitochondria, our chloroplasts also have DNA inside of them, which are highlighted in here, or shown in the little blue spheres. Okay. So again, the DNA is compartmentalized in chloroplasts. So the chloroplasts don't need nuclear DNA to regulate their processes. So this is a really cool feature of chloroplast and mitochondria. I'd like for you to identify, maybe you want to draw this too on your notes when you're studying, is the space surrounding the thylakoids is called stroma. It looks void here, but I mean there's liquid in there, and that's called the stroma. The stroma is the innermost liquid-filled area of a chloroplast. Okay. In all of these structures that we just identified, are membrane-bound, including the nucleus, the smooth and the rough ER, the Golgi apparatus, lysosomes, peroxisomes, vacuole, mitochondria, and chloroplasts. Okay, they all fall under a category of an organelle because they have a membrane around them. There's a really neat website from the University of Utah. They have the Learn Genetics website. If you go to the link, where is it? I think it's learn.genetics. Oh, here it is. learn.genetics.utah.edu. And go in there and go to the cells, Amazing Cells link, and then you can go into the inside of the cell and then also look at the membranes, right? So there's some really cool study tools for you to help put this into perspective for you. So check out that website for an additional study tool. In addition to membrane-bound structures being of use to a cell, there are also non-membrane-bound structures that work for a cell. One important feature that I'd like to address are these things collectively that comprise the cytoskeleton. The prefix cyto refers to cell, and the skeleton, right, it gives scaffolding. There are three classes of proteins that make up the cell cytoskeleton, which include microtubules, intermediate filaments, and microfilaments, microtubules, and intermediate filaments. And working left to right, in the image over here, the microtubules in the far left column, microfilaments in the middle column, and the intermediate filaments over here on the right hand side. So let's just get an idea about what these things do. This next image just illustrates how dispersed they are, how they are within the cell cytoplasm, but certainly they don't have a membrane around them. The first cytoskeletal element is called a microtubule, and focus on the end of the word, the tube part. These form a tube inside when they're arranged, and what they do is transport vesicles, like we saw between the rough ER and the Golgi, or from the Golgi apparatus to the membrane. Okay, they physically aid in transport of vesicles. Or you may remember from the video that I showed earlier, the little vesicle being transported along. That thing carrying it, what it was walking along, was a microtubule. Microtubules are also really important for structure of these features called cilia and flagella, which provide movement to an entire cell or allow movement of liquid substances across a cell surface. You can see some examples over here on the right hand side. Lower right, the little yellow projections that I'm going to try to highlight in red. Those are called cilia. You would find these, for example, lining the respiratory system in your body, or females lining the female uterus to allow for fluid movement to keep, say, an oocyte moving right down the reproductive tract. Okay. And in the far right, we have some sperm cells. You can see this long extension I'm going to highlight in red will be a flagella. Cross section of them, if you look really closely, you can see multiple microtubules help making these big structures. Okay. And so, another important function of microtubules that we'll use later is during cell division, when our chromosomes are pushed and pulled within the cell, microtubules are doing this work. Okay. Of the cytoskeletal elements, the microtubules have the greatest diameter, okay, and they also do form a tube, and the protein inside of them is called tubulin. Okay, that's just the name of the protein. They are hollow and kind of slender and elongated. Okay. That is a summary of the microtubules. Here's a nice image of a microtubule facilitating or aiding in vesicular transport. You can see the microtubule in the purple structure. You can see how it is hollow in nature, how they get the term tube. And then on the top you can see a vesicle in the orange, and then there's a set of proteins called motor proteins that actually transport the vesicles, right, in one direction or the other. Okay, and here's a couple of alternative images of cilia and flagella. Okay, in the top left are a bunch of tiny cilia, and on the far right a single flagella for, in this case, a sperm cell. Another type of cytoskeleton component is called a microfilament. Don't get confused because they both start with the two that we've talked about have the same prefix, micro. Again, focus on the end, the filaments. The filaments form filaments that look straight. The microtubules form tubes. Okay, so just focus on the end of the word instead of the first of the word. These are really important in muscle contraction in your skeletal muscles, and they also give physical structure support to some cells, like in your digestive system. You have these projections called villi that project out to increase surface area to promote increased absorption, and so these projections stick out due to these things called microfilaments inside of the cell. These have the smallest diameter of the three filaments, and the proteins that make up the microfilaments is called actin. Oh, and I need to remove this little box right here. They are not hollow nor slender. I forgot to remove that. Fix this really quick. Thank you for your patience. Okay, so our microfilaments, their smallest diameter, they're made of actin, and they are thin, and they form, as you can kind of look over here, just straight chains basically, or a filament with one another. So microtubules, microfilaments, and then here's an example of how our microfilaments can aid in cell motility. There's a white blood cell. It almost looks like a creature, but it's a white blood cell, and you can see the plasma membrane extending out, and this extension right here and splaying out is due to inside actin proteins, right, working to push the plasma membrane. So the cell looks like it's walking because of little proteins pushing its membrane out. And then what it's doing is it's going towards these bacteria, and it's going to try to attack them and get them away and kill them. The next type of filament is called an intermediate filament. Nothing fancy about its name. It's intermediate in size. It's not the biggest like the microtubules. It's not the smallest like the microfilaments, but it's intermediate in size. Some important primary role of the intermediate filaments is to give physical protection to cells that would be exposed to physical, like stress. So like your skin cells, you think about how much tensile strength they're always being exposed to. So even when you just put on your shirt, you're rubbing the shirt across your skin, or your rings, or your hats, your glasses. That physical touch could actually be damaging and separate the cells. And so to prevent those cells from breaking apart, those types of cells would have intermediate filaments. An important or a well-characterized type of intermediate filament is called keratin. It's not the only one, but if you look in the image on the far right, it does form filaments, as the name implies, and they interlace to give physical strength to that particular cell type. Okay. So collectively, these things are called the cytoskeleton. And as well, we cannot forget about the role of ribosomes. They are not part of the cytoskeleton, but they're another feature common to all cells that aid in, as we identified earlier, protein synthesis. Okay, these are critical for the manufacturing of proteins. Okay. If you look at the basic structure, the ribosome has what's called a small and a large subunit. And if you look at the image on the right-hand side, you can see where the small and the large subunit are actually separate. When the ribosome is not working, the small and subunit are not complexed or joined together, so they would physically be separate like this, and may not even be that close to one another. But when they are making proteins, the large and the small subunits would join together, like you can see in the image to the right. Okay, what a ribosome is, it is protein, so it's basically a globular protein, but it also has RNA inside of it. And that, when we get into the third unit, we'll look at how that RNA actually aids in protein synthesis. Okay, so ribosomes, not technically an organelle, so that's why we have in this other list, but really critical to protein synthesis. We will find them either dissolved in the cytoplasm to make proteins that work inside of the cytoplasm, like we just listed. The microtubules, microfilaments, etc. Those are proteins that would have been manufactured by ribosomes free in the inside of the cell. Conversely, we learned already that ribosomes can attach to the rough ER, and when they work with the rough ER, they're making proteins that will be secreted or incorporated into a membrane somewhere. Okay, those are the ribosomes. Another feature unique to a plant cell is going to be the presence of a cell wall. A cell wall is not the same thing as a cell membrane. A plant cell will have a plasma membrane or a cell membrane and a cell wall. It's easy to kind of get the terms confused, but I want you to make sure you do not confuse a wall versus a membrane. What the plant cell wall does is give physical structure and support to the plant. And if you think about it, your plants don't have a skeleton like, say, you do. Okay? They don't have the ability to be upright in the environment, and so they need to have something extra to allow for them to kind of be supported in their environment. And they have this extra cell wall. What it is a complex polysaccharide, like we studied in Chapter 2. So it's just a big sugar group all joined together, giving physical strength to it. Okay, but it is rigid. However, you will find these things called plasmodesmata between individual cells, so that each cell is not completely walled off from another. There's little gaps. Okay, I'll try to highlight over here between individual cells. Okay, it's called a plasmodesmata, and it's just the physical gap so that the fluid of one cell is connected with the fluid of another. And then the cell wall itself is illustrated in the green colored area. Before I move on to animal cell junctions, I want to go back and point out a few things about an animal cell versus a plant cell. Okay, characteristically, right, what makes a plant cell is the presence of chloroplasts. Okay, as well as the presence of a central vacuole, and now we know the presence of a cell wall. Okay, what I don't want you to get confused is that just because plants have chloroplasts or central vacuoles doesn't imply that they don't have everything else. They do. Plant cells have mitochondria, just as do animal cells. They have a membrane, just like animal cells. They have a liquid cytosol. They have a complex cytoskeleton. They have ribosomes attached to membranes, ribosomes free in the cytosol, etc., etc. But they have extra features, including the chloroplast, the central vacuole, and this outer cell wall. Okay. And as I started out this chapter discussion identifying that, we'll conclude this chapter looking at how animal cells can adhere to one another. We have these specialized junctions which aid in tissues being joined or being formed. If you look at the image from your textbook, on the far left you can see 1, 2, 3, 4 cells joined together. Then you can see the proteins joining them together, which are going to form these junctions. Okay. The different types of animal cell junctions include what are called tight junctions, adhering junctions, and gap junctions. The tight junctions, as their name implies, form a tight junction between two cell membranes, so that the two membranes are physically connected with one another, and basically it just attaches two cells, and where there are tight junctions there is very minimal space in between the cells, and then therefore the likelihood of a molecule moving between two cells where there's a tight junction is very minimal. It's like almost not going to happen at all. Right? So a tight junction you can see illustrated on the first set of images on this image to the right hand side. You can see the little purple dots. Those aren't purple dots, they're representing proteins, and the two yellow things are the cell membranes of two different cells, so the cells are joined together. Another type of junction is called an adhering junction, and that allows for two cells to bind together like what we saw described in a tight junction, But if you look at the proteins in this image, you'll see how the proteins actually interlace. And I actually like this image because it almost looks like two hands interlacing. So you could take your two hands and interlace your fingers and then try to squeeze them together tightly, your fingers tightly, and then try to pull them apart. And you'll notice when you try to do that, when your multiple fingers or multiple proteins interlace, it gives physical strength to that junction. You should also note that not only are there proteins interlacing between the cells, but the lacing continues inside of the cell and attaches to the intermediate filaments of the cytoskeleton. So where you find an anchoring junction is in parts of cells where they're likely to rip apart because they're physically doing some mechanical activity. Okay, so like for example, in your heart cells. If you think about what the heart cell does, it's always beating and generating force, and so therefore there's so much tension being generated between individual heart cells. I mean, if there were no adhering or desmosome junctions, the heart tissue would actually rip apart, which you know is not good. Okay, so those are there to distribute tension so cells don't rip apart. And the third type of animal cell junction that's common is called a gap junction. It too forms a junction between two animal cells. It is also made up of proteins, but if you notice that the proteins, they're not aiding in any type of tension distribution. And then if you look really closely, you'll see a channel that goes between the cell types. Okay, and so what this does is allows for molecules to diffuse between one cell to another. Okay, and where this is common is, for example, in your in the smooth muscle in your skin, not in your skin, excuse me, the smooth muscle in your digestive system. There's a hole, or the gap junctions allow for ions like sodium and potassium to move from one cell to another. And if you remember, ions have charges, so basically it allows for an electrical impulse to flow from one cell to another. Okay, it's a form of communication. Also in your heart you have a lot of gap junctions, so electricity can flow from one cell to another. So in summary, those are our different types of animal cell junctions. It concludes our Chapter 3 discussion of different cells and eukaryotic cell functions. All right, we'll see you online or in class.